Hello, this is Dr. Oviedo. Today I want to discuss remdesivir, including the mechanism of action, and I also want to discuss clinical trials. What is remdesivir? It's a nucleotide analog. It's a prodrug that undergoes metabolism to an active form. In this form, remdesivir blocks RNA polymerase, which the virus uses for replication. Remdesivir was originally developed as a potential treatment for Ebola virus. This is the chemical formula of the active form of remdesivir. For today's discussion, I'm going to use this pink form to represent remdesivir. Before we discuss the mechanism of the drug, I would like to remind you how COVID-19 infects a type 2 pneumocyte, which is a very important cell which lines the alveolus in the lung. Here's our virus. Here I've shown it attaching to ACE2, which is the receptor for COVID-19. Once it's attached, it can then enter the cell and release its RNA. Once inside the cell, the RNA can make more viruses. This here represents the remdesivir. The remdesivir can block the viral RNA from producing more viral RNA. Before we discuss the mechanism in more detail, I want to show how the virus uses viral RNA polymerase to make more viral RNA. Here I've shown the COVID-19 RNA. This contains instructions to make more virus. If you look at it very carefully, you will see that it is actually composed of multiple nucleotides that make up the RNA strand. I'm going to take a closer look at this portion right here. Here I've shown a string of nucleotides which represent a portion of the viral RNA. This is viral RNA polymerase, which is a molecule that can make more viral RNA. Let's see how this works. These blue dots all represent nucleotides present in the cytoplasm. The viral RNA polymerase will use it to make more viral RNA. You can see it moves forward as it adds the nucleotides. Once it's added enough to make a full portion of viral RNA, it can then go ahead and package it inside a virus. That virus, of course, can go on to infect other cells or other people. Now let's go on to see how the remdesivir works. Here I'm going to show how the viral RNA polymerase is blocked by remdesivir. Here again we have our viral RNA, and here is our close-up. This time, in addition to the nucleotides that the viral RNA polymerase needs, we also have a few molecules of remdesivir. Let's see what happens. You can see the viral RNA polymerase has added 2 nucleotides. However, this time the viral RNA polymerase has added a remdesivir molecule instead of a nucleotide. This will block the viral RNA polymerase from any further activity, and there is no more virus. At this point, I want to discuss the principles of clinical trials along with clinical trials done in humans with remdesivir. A phase 1 clinical trial is done after extensive detailed studies in the lab, including animal studies. The purpose of a phase 1 trial is to determine safety, toxicity, dosage, and pharmacokinetics. Phase 1 clinical trial is designed to find out how the drug will behave when administered in a person. These studies have already been done with intravenous remdesivir when it was being studied for Ebola. Those studies did show that there is some kidney and liver toxicity in remdesivir. However, it was considered a good enough drug to go on to phase 2 studies. The purpose of a phase 2 clinical trial is for efficacy. We want to answer the question, does the drug have an effect on patients with the disease? This has already also been done in patients who had Ebola infection. Because phase 1 and 2 clinical trials had already been done with remdesivir, we were able to begin with phase 3 clinical trials. This saves an enormous amount of time. Phase 3 is a randomized clinical trial. We are comparing the drug under study to another drug or a placebo in limited numbers of patients. These patients are given either the drug or the placebo to see if there is an effect. This is where we are with Remdesivir and COVID-19. If the phase 3 trial shows that Remdesivir has a beneficial effect in patients with COVID-19, it can then go to the FDA for approval. The phase 3 clinical trial would have to show that patients that received the drug did better than patients that did not receive the drug. If a drug is successful in phase 3 clinical trials, it can then go on to phase 4 clinical trial. In a phase 4, we continue to monitor this drug in more patients after the FDA approval. This is very important to determine long-term safety and effectiveness and to learn about adverse events that may happen in larger populations than those that were studied in the phase 3 trial. So why are clinical trials important? Does the drug do what we think it does? Does the drug produce adverse events that we would like to know about? Are there different effects in different populations? Is the drug safe for all populations? Is the drug more beneficial than current therapy? There is no current therapy for COVID-19. The drug, however, has to be more beneficial than supportive therapy, which is what we are currently doing for COVID-19. Supportive therapy means we do things like intravenous fluids, ventilation, in order to help support a patient as their own body fights the virus. I would like to discuss the reference I used for today's presentation. This JAMA paper from April 13th, 2020 discusses numerous potential drugs for COVID-19. This April 15th, 2020 paper describes the drug mechanism of action in Ebola, SARS-CoV, SARS-CoV-2, and MERS-CoV. Okay, that's it. I am really looking forward to the results of the clinical trials for Remdesivir. Please do everything you can to stay healthy.